Hello, welcome. By learning, unlearning and relearning together, in the next few minutes, you'll be able to clear your conceptual knowledge of cell wall and plasma membrane, which will give you a solid platform to excel in your exams. Today, we will learn together about cell wall, its components, its structure, differences between primary cell wall and secondary cell wall, and also functions of cell wall. In addition, we'll study plasma membrane, fluid mosaic model, structure of plasma membrane, role of protein molecules, transport across plasma membrane, and certain terms like endocytosis, exocytosis, phagocytosis. In our previous session, we had learnt about cell in detail. If you haven't watched that video, I strongly recommend you to watch that video first and then proceed with this video. You can find the link of that video in the description below. As you very well know, cell wall is present only in plant cell and absent in animal cell. It is the outermost covering present adjacent to the cell membrane in plant cells. Its main function is to provide strength, structural support, and protection to the cell. We will learn together about its various functions a little later. Now, we will study about the components of cell wall. The cell wall of plant cell is made up of a fibrous polysaccharide, namely cellulose. What do you understand by the term polysaccharide? Polysaccharides are complex carbohydrates made up of hundreds or thousands of simple sugar units. Cellulose molecules are arranged parallel to each other and are joined together with hydrogen bonds. The bonds between cellulose molecules are very strong and difficult to break. Cellulose makes plant stems, leaves and branches strong. Next we come to bacterial cell wall. It is rigid and is made up of Peptidoglycan or murine, consisting of sugar and amino acids. Muramic acid is a typical cell wall component of bacteria. Many molecules of peptidoglycan join together to form an orderly crystal lattice structure. It gives strength to the outer surface of the organism. Fungal cell wall is complex and flexible structure composed of a glucose derivative known as chitin, another glucose polymer namely glucan, and hydrophobin proteins which give strength to the cell, helps adhere to surface and control the movement of water into cells. Let's study the structure of cell wall with the help of diagram. You can clearly see a multi-layered structure identified as middle lamella, primary cell wall, secondary cell wall. Middle lamella connects two adjoining cells. Primary cell wall is found between middle lamella and plasma membrane. Secondary cell wall is found between primary cell wall and plasma membrane. At certain places, the secondary wall shows the presence of pits. The pit membrane is permeable and allows passage of materials. Next, the middle lamella is made up of polysaccharides, namely pectin, consisting of calcium and magnesium pectate, which helps adjacent cells to bind or adhere to one another. Primary cell wall is composed of cellulose, hemicellulose and pectin. We have already discussed function of cellulose. Biological role of hemicellulose is their contribution to strengthening the cell wall by interaction with cellulose. Pectin contributes to complex physiological processes like cell growth and cell differentiation. The primary cell wall gives strength and flexibility for cell growth. It is thin, elastic, and capable of expansion in a growing cell. Secondary cell wall is rigid layer, giving strength and support to the cell. It is composed of cellulose, hemicellulose, and lignin. Cell wall matrix contains lignin and suberin. Let us find out the difference between primary cell wall and secondary cell wall. The primary cell wall lies inner to the middle lamella. The secondary cell wall lies inner to the primary cell wall. The primary cell wall is formed in young growing cells. The secondary cell wall is formed in a mature cell. Primary cell wall is single layered, while secondary cell wall is generally three-layered. It is elastic, capable of expansion. This is regarding primary cell wall, whereas the secondary cell wall is rigid, incapable of expansion. It is 1 to 3 micrometer thick. This is again regarding primary cell wall. 5 to 10 micrometer thick is secondary cell wall. Growth in primary cell wall is by intussusception. In the case of secondary cell wall, growth in thickness is by accretion. Let me explain both the terms. The primary cell wall grows by addition of more wall material within the existing one. Such a growth is termed intussusception. In mature cells, more layers of cell material are added internally to the primary wall. These layers are called the secondary cell wall. Growth by addition of cell wall material on the existing one is called accretion. Next difference is, in the case of primary cell wall, cellulose content is relatively low but high in the case of secondary cell wall. Primary cell wall lacks additional material. Secondary cell wall shows the presence of lignin and suberin. Pits are absent in primary cell wall. Pits are present in secondary cell wall. Plasmodesmata are formed. These plasmodesmata are cytoplasmic connections between cells. Now, we are coming to the function of cell wall. It provides mechanical strength and support to the cell. The cell wall maintains shape and form of the cell. It also protects the cell from mechanical injury. It acts as a barrier against pathogens. It also allows movement of materials in and out of the cell. Chitin and suberin deposits check loss of water from the cell surface by evaporation, and pores in the cell wall permits plasmodesmata to connect all the protoplasts. So we have studied the structure, composition and functions of cell wall. Next, let's take up plasma membrane. Plasma membrane is a biological membrane which forms a stable barrier between two adjacent compartments. It is also known as cell membrane, plasma lemma, cytoplasmic membrane. Now let's study fluid mosaic model with the help of diagram. This model was proposed by Singer and Nicholson in 1972. Which are the parts prominent in the diagram that is shown to you? Phospholipid layer, peripheral proteins, integral protein. This matrix that is highly viscous has two layers of phospholipid molecules having globular proteins associated with them. Coming to phospholipids, the polar heads of the phospholipid molecules are directed outward and non-polar tails pointing inward. It forms a water-resistant barrier which allows only lipid materials to pass through it. Proteins are of two types: peripheral or extrinsic proteins, integral or intrinsic proteins. The peripheral proteins are loosely attached to the outer side of the cell membrane. The integral proteins are embedded in the phospholipid matrix. Some integral proteins have very large molecules and project on both the surfaces. These are called tunnel proteins or transmembrane. Water-soluble materials pass through these channels. Some integral proteins that have small molecules lie in the outer or inner layer of phospholipid molecules. Carbohydrates or oligosaccharide molecules present in the exposed surface of the cell membrane are associated with proteins as well as lipid molecules. They help in cell recognition and cell binding. Let's pause for a moment to revise and relearn the concept that we have learned so far about structure of plasma membrane. Plasma membrane is made up of phospholipids, proteins, carbohydrates. The proteins are further grouped into peripheral or extrinsic protein and integral or intrinsic protein. Next we come to role of protein molecules. The protein molecules of the cell membrane may act as carrier molecules, receptor molecules, and enzyme molecules. Carrier molecules: the carrier protein molecule known as permease binds with the specific molecules and help in their transport. Coming to receptor molecules, the glycoproteins on the cell surface act as receptors. For example, insulin binds to the cell membrane receptors to produce their effect in the cell. Enzyme molecules: these molecules are present in the plasma membrane to carry out their functions. For example, enzymes for respiration, ATP synthase, are present in the inner membrane of mitochondria. Next we come to transport across plasma membrane. Plasma membrane is said to be selectively permeable because it allows certain substances to pass in and out of the cell. This takes place broadly by two processes: physical processes and biological processes. Biological processes consists of transport by membrane proteins, It may be passive transport by channel protein, passive transport by carrier proteins, or active transport by carrier proteins. We will discuss diffusion and osmosis first. The normal tendency of a substance to uniformly spread from a region of higher concentration to a region of lower concentration due to its kinetic energy is known as diffusion. It helps in the respiratory exchange of gases between cells and their surroundings. In plants, it also helps in transpiration. Osmosis is the diffusion of water or solvent through a semi-permeable membrane from a solution of lower concentration of solutes to a solution of higher concentration of solutes. It is through osmosis that the roots of plants absorb water from the soil. Osmosis also brings about movement of water from cell to cell. Since there is no expenditure of energy during diffusion and osmosis, these modes of transport are said to be passive. Diffusion of oxygen from lungs to capillaries in human is an example of passive diffusion. Reabsorption of water from kidney tubules is an example of osmosis seen in human excretory system. Now we come to biological process. First, passive transport by channel proteins. These proteins do not bind to solute molecules. They help in the passive transport of molecules by forming an aqueous channel. Passive transport by carrier proteins. Some carrier proteins bind to specific solute molecules and passively move them through the membrane down their concentration gradient. Active transport by carrier proteins. Some carrier proteins bind to specific solute molecules and with the help of energy move them through the membrane against the concentration gradient. Since energy is required by this process, it is said to be an active transport. Next, we come to certain processes that occur within a cell, namely endocytosis, exocytosis and phagocytosis. Endocytosis, as seen in the diagram, for the intake of particles, first the plasma membrane forms an invagination. It forms an intracellular membrane-bound vesicle for surrounding the particle, mainly for nutrition. Coming to exocytosis, a cell gives out secretions by exocytosis. First, an exocytotic vesicle containing the secretion comes near to the plasma membrane and then fuses with it. An opening is formed as shown in the diagram and the secretion or the contents are discharged. In human beings, for example, the secretions discharged from gland cells, namely digestive enzymes, are secreted by exocytosis. Next, we learn about phagocytosis. You can see in the diagram, a cell takes up a large particle by surrounding it and engulfing it. In human beings, for example, the leukocytes or white blood cells, that is WBC, present in the blood protects us by ingesting microbes that have entered our body. It is then engulfed by phagocytosis. Let us go through the comparison between cell wall and the plasma membrane. First, cell wall is present in plant cell bacteria, fungi. Plasma membrane is present in all cells. Cell wall is non-living, hard and rigid, while plasma membrane is living, soft, flexible. Cell wall is completely permeable. Plasma membrane is semi-permeable. Cell wall is made up of cellulose, chitin, pectin. Plasma membrane is made up of phospholipid, protein, carbohydrate. Cell wall do not possess receptors. Receptors are present in plasma membrane. Cell wall protects cell from external environment, while plasma membrane, in addition to protection, it also maintains the internal environment. I'm sure the concept taught is clear to you. Finally, I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.